Okay, so if you plot this heat map for the other genes selected, you should have found that the highly variable genes, quite a large number of those are just sort of generally noisy and are not expressed in one specific cell type, whereas the highly correlated genes should have looked just as good as these M3 drop genes and are quite highly associated with the different cell types. And the PCA genes, it will depend on which principal components you chose, how well they were associated with the different cell types or not. And now we're going to move on to pseudotime. So pseudotime is the sort of alternative to clustering when thinking about your data. So when we're thinking about clustering, we think that there's distinct groups of cell populations in our data set that are quite clearly different from each other. Whereas in pseudotime, we're assuming that there's some sort of gradient within our data set where the cells are smoothly transitioning from one biological state to another biological state or multiple other biological states. And so for pseudotime, we want to fit some sort of path or trajectory or multiple branches to this smooth transition. And the reason we can't just use sampling time or some sort of time series is that, in general, single cells will differentiate at different rates compared to each other. So if you take a sample at time 0, time 1, time 2, time 3, cells in time 2 or 3 might be at different stages in the actual differentiation process or whatever smooth transition you're looking at. Thus, we want to sort of infer this underlying path and then place the cells along that path. And of course, we can't do an actual time series properly with one individual cell because every time we do single cell RNA-seq, we have to destroy the cell. There are many different tools for doing this, and there's this good review or summary of the various methods that were available in 2016, but there are new ones since then as well. It seems every couple of months somebody comes up with a new method for inferring this. So do check the literature for the latest methods and if there's any good comparison yet. So these are some of them. They're written in different languages. They have different pluses and minuses to them. We're going to look at ones that run fairly quickly and can be run in R rather than sort of Wanderlust and Scuba, which are in MATLAB. And we're going to avoid ones that didn't have proper documentation when we were writing the course. Can focus on ones that looked fairly finished and had proper documentation. So to do pseudotime analysis, almost all of these methods use multiple steps to do this. First, you have to define some sort of similarity. So first off, you may or may not have to do feature selection before you do pseudotime analysis. So for Monocle, requires you to do feature selection because it's too slow to run on all 10,000 genes, whereas other ones like Destiny, which is not in this figure, are fast enough that they can run on all of the genes in your matrix. So you have the choice to do feature selection or not. Then you have to calculate some sort of similarity or distance between your cells, and there are different ways of doing this. So cosine similarity has the advantage of correcting for library depth effects, whereas Euclidean distance does not. So you would have to very thoroughly normalize your data before applying a method that uses Euclidean distance. Then there's usually some step involved in dimensionality reduction. So it might use principal component analysis or t-SNE for dimensionality reduction. Or you might use diffusion maps for pseudotime to find lower dimensions for your data. Some methods will cluster your data before they try and join those clusters together with a pseudotime trajectory. Others will find the trajectory for single cells. So there's a bunch of different ways of doing clustering. Most of the methods will then use some sort of graph to plot paths through your cells to identify that sort of high density region, and then find the paths through that graph and identify the longest path or the most important path as the pseudo-time trajectory for your cells. And then have some sort of method of ordering, projecting all of your cells down onto that path to order them through pseudo-time. And then usually during this path-finding step is when you'll also identify multiple branches if they exist, based on different metrics of how different your paths are from one cell to another, based on where you are in the overall data set. So all of these methods also have other pluses and minuses to them, so they might be unbiased or they might not be. Some are scalable with respect to genes, some are not. Many are scalable with respect to genes, but some are not. Most of them are quite well documented at this point, and many of them require extra parameters that you'll have to define a priori, and some of them do not. So I'm going to quickly go through the methods we've chosen to look at that sort of cover different ways of doing pseudotime analysis. So T-scan is a clustering-based pseudotime analysis method. So it first finds clusters using mClust, which is very similar to sort of k-means. And then it builds a minimum spanning tree connecting all of those clusters to their nearest neighbors, essentially, and then looks for whether there are branches in that tree, and then finds the longest branch, and that becomes your pseudotime trajectory with side branches splitting off of them. So we're again going to use the Deng data set for this. And then, so for T-scan, first you have to pre-process the data. Then you do the clusters, and you have to specify how many clusters are in your data set. It won't figure that out for itself. Then you can plot the result. And here you can see it's colored the different clusters it's found, and then these lines connect the clusters together. In this case, it didn't find any branches. It's only a single trajectory. And here's how you get the single cell order out of here. And then we can, so here we can look at which cells have been assigned to the different clusters that T-scan identified. So here we've got mostly 2 cells in cluster number 10, and different clusters will contain different cells. Here I've plotted the inferred pseudotime order versus the actual time point in order. So you can see T-scan didn't do a very good job because it has, it's reversed everything, because most pseudotime methods don't know which end is the start and which is the end. So quite often they'll pick the end as the start and the start as the end, which is what T-scan has done. And it's also made this mistake of thinking the blastocyst cells come in between the 4 cells and the 8 and 16 cells. Yep.
Does this can use raw normalized data?
This uses raw data, I believe, because it applies its own normalization. No, this uses normalized data. Different methods use different types of data. So T-scan uses normalized data because we're using log counts here, whereas I believe Monocle uses raw counts. So we're going to move on to Monocle. And as mentioned, Monocle requires you to do feature selection. And it does not use clustering. It directly builds multiple minimum spanning trees on a reduced dimension representation of the cells, and then identifies the longest path in the tree to determine the pseudotime. And the current version of Monocle will actually fit multiple different trees with different numbers of branches to try and figure out what the best number of branches are for your particular data. But you have to do feature selection first, so here we're just using M3-drop feature selection, which we just talked about. And then we can run Monocle. So knowingly, Monocle has their own bioconductor class, so you have to create an object in their particular class to be able to use it. And here we set the features we want to use. And here Monocle asks you to provide raw counts, and it will do its own feature selection. It will do its own normalization, so you have to estimate the size factors for its own normalization, then reduce the dimensions, and then order the cells to find the pseudotime, and then you can plot it. So Monocle has actually found 2 branching points for these little tiny branches here and here, which are probably incorrect. But the overall trajectory, it's fit quite well. And this is how we can extract pseudotime order from Monocle. So here's where it's stored the so here's where Monocle stores its pseudotime. And we can plot it against the actual time points, and you can see it's quite accurate in this case, but it's gotten it reversed again. The last method is looking at diffusion maps. And diffusion maps basically find a nearest neighbor network by modeling diffusion from each cell to every other cell in the data set, and then infers a low-dimensional manifold by estimating the eigenvalues and eigenvectors of the diffusion model it's inferred between the different cells. And this is in the Destiny package. So diffusion maps requires should be used log-transformed normalized data. And then you can just extract the top 2 eigenvectors to find the diffusion components. You may have to look at 3 or 4 depending on how complex your data is. And if you plot that versus the cell type, the known sampling time point, the early cell types are quite clear, but then the later cell time points are all sort of overlapping. I'm going to skip the exercises for now, and you can work on them after lunch. And the last method we're going to consider is Slicer. Quite similar. So it automatically selects its own features, so you don't have to do your own feature selection. And then it uses, again, a neighborhood-based method to do that. Yeah, and it uses a nearest neighbor network to then identify the connections between cells and then paths in that network to identify the pseudotime. So here you have to identify what k you want to use for your k nearest neighbors. So how many cells should every other cell be connected to in the graph? And it provides a method for identifying the optimal k. We have to set a k-min or a k-max because it can have strange behavior for the select k, where in certain times it will select the wrong k if you allow it to go from a too low value of k. It'll select a very low value of k and then find nonsense at the end. And then it does dimensionality reduction. This is the dimensionality reduction method. And then we can plot its inferred lower dimensions. And see again, these early time points are quite well identified, but the later time points are a bit of a mess. You can also plot the k nearest neighbors graph from Slicer, so you can see it's inferred a single trajectory. And after you've plotted, after it sends this lower dimensional representation, you have to find the two ends of the continuum of cells using find extreme cells. And here you can use this to then select which end should be the start and which should be the end of those 2 points. So we've told it that this one is the start using this command, and then you can tell it to order the cells using that start point. And then assign branches if you're looking for multiple branches. And we can plot this, and now we've got the right order from 2 cells all the way to blastocysts. And very quickly, just look at, and then you can compare the different methods against each other, see how much they agree or not. And each of the methods have a way of plotting the expression of different genes through time. Here's just an example for T-scan and how you do that for monocle for whatever particular gene you're interested in. And we're going to break for lunch. And then for an hour, so be back here at 1:30, and we'll give you some time to work on the exercises for pseudotime, and then cover the remaining 5 sections, which should take us till about 3 in the afternoon.